Hey everyone, and a big shout out to Mr. Martin's IB Bio class out there.
We heard you guys are diving into carbohydrates and lipids.
Yeah, those are some pretty essential molecules in biology.
Absolutely essential.
And I bet you're probably feeling a little bit overwhelmed with all those notes and textbook chapters, right?
It can definitely be a lot to take in.
So think of this deep dive as you're like a cheat sheet, you know?
Yeah, we'll break it all
We're
down.
going to break it all down into like the need-to-know info on carbohydrates and lipids. And by the end of this, you'll be able to explain all the key structures, the functions, the differences.
And we'll make sure to keep it simple.
Yeah, no need to be a chemistry whiz here.
Exactly. It all starts with that element, carbon.
Carbon?
Yeah, it's like the ultimate building block of life. And you know what makes it so cool?
What?
It's so versatile. It can form 4 bonds with other atoms.
So it's like the social butterfly of the periodic table.
I like that analogy. Yeah. Always making connections.
Always making connections.
And those bonds, they're called covalent bonds, and basically it means that the atoms are sharing electrons.
Like holding hands at the atomic level. I love it.
Exactly, and since carbon can actually bond with other carbon atoms, they can create these long chains or even rings.
So it's like Legos?
Yes, exactly, like Legos. Carbon is that brick that can connect in all sorts of ways to build all these amazing structures.
So carbon is our master builder.
Yeah.
But how do we go from like a single carbon atom to these giant molecules like carbohydrates and lipids?
That's where these things called condensation reactions come in.
Condensation reaction.
Yeah, it's kind of like building that giant Lego castle, right? To join those smaller pieces, those monomers.
Monomers.
You got to remove a water molecule, H2O.
Oh, so it's kind of like a dehydration process.
Exactly. You're
Yeah.
taking out water to link those things together, and that creates a special bond and links those monomers to form bigger structures called polymers.
Okay, so dehydration to build, and then what about breaking down?
Breaking down.
Yeah, like when we digest food.
You're thinking about hydrolysis.
Hydrolysis.
Yes, that's the opposite of condensation. Instead of removing water, we add it back in to break those bonds.
Got it. So it's like the reverse process.
Exactly, and that's how our bodies take those large food molecules and break them down into smaller bits that our cells can actually use.
Makes sense. Okay, so we've got our carbon, our master builder, creating these long chains. Now let's zoom in on carbohydrates.
Carbohydrates.
Yeah, I think we all know sugar's a carbohydrate.
Of course.
But what exactly are they?
So sugars are the simplest form of carbohydrates, or we call them monosaccharides.
Monosaccharide.
Yeah, they're like the basic building blocks. They're single sugar units, and they can't be broken down any further.
Okay.
And there are different types, but let's focus on glucose.
Oh, glucose, the rock star.
Exactly, the rock star of monosaccharides.
But why is it so important? Everyone's always talking about glucose.
Well, imagine glucose as like the fuel that powers your cells.
Okay.
It's got the special formula C6H12O6.
I've seen that before.
But what makes it really special is its structure, glucose, is polar, which means it dissolves really easily in water.
Oh, like when you put sugar in your tea or coffee.
Exactly, it just disappears.
Yeah.
And that means it can travel easily in your blood to reach all your cells.
Okay.
And the best part, it releases a ton of energy when it's broken down by your cells through this process called cellular respiration.
So it's like the energy drink for ourselves.
I like that analogy.
But here's where I get a little bit tripped up. I've heard that glucose has like this split personality or something, alpha and beta glucose.
You're right, glucose can actually exist in different forms. We call them isomers.
Isomers.
They have the same chemical formula, but they have a slightly different arrangement in space.
So it's like having all the same Lego pieces, but you can build different things with them.
Exactly. And alpha and beta glucose are 2 key isomers, and the only difference is the orientation of one tiny part on the molecule.
Wait, so just by flipping one tiny part, it makes a difference?
It makes a huge difference because that tiny change impacts how the glucose molecules link together to form larger carbohydrates, those polysaccharides.
Polysaccharides.
Yes.
Okay, so that's where we get into like starch and cellulose.
Exactly. Polysaccharides are long chains of glucose all linked together.
Okay.
Take starch, for example. It's how plants store their energy.
It's like the plant's pantry.
Exactly. Their pantry full of energy.
And then there's glycogen, right? The animal version of starch.
Yes, glycogen is how we store energy in our muscles and liver ready for quick access.
Like an energy reserve.
Exactly, an energy reserve.
And what about cellulose? That's what gives plants their structure, right?
Yes, cellulose, think like the wood in trees. It's incredibly strong because of how those glucose molecules are linked.
Wow.
They form these long, straight chains that create tough fibers, perfect for building those plant cell walls.
So just by changing the arrangement of glucose, you get completely different molecules with totally different jobs.
It's amazing, isn't it?
It's amazing. Okay, so we've got starch, glycogen, cellulose, all made of glucose, but very different structures and functions. What other cool things can carbohydrates do?
Well, they can actually team up with proteins to form these things called glycoproteins.
Like a protein.
Glycoproteins.
What are those?
Think of them as proteins with little carbohydrate decorations attached.
Decorations.
Yeah, and these decorations, they act like name tags for cells, helping them recognize each other.
So, like, if a cell with the wrong name tag shows up, the body knows something's off.
Exactly. A great example is our blood types.
Mm-hmm.
The ABO system, different blood types have different glycoproteins on their red blood cells.
Right.
And that's why receiving the wrong blood type can be dangerous. Your body sees it as an invader and attacks.
So thanks to carbohydrates, our bodies can tell friend from foe at the cellular level. That's pretty amazing. Okay, we've explored the world of carbohydrates from simple sugars to complex structures. Now let's switch gears and dive into the world of lipids. What are these guys all about?
Lipids are a diverse groupthink: fats, oils, waxes, even some hormones. But what unites them is that they're hydrophobic.
Hydrophobic sounds like they don't like water.
They don't mix well with water. Think about oil and water separating in a salad dressing.
Right.
That's because lipids are mostly made of carbon and hydrogen forming nonpolar bonds, but water is polar, and nonpolar molecules just don't mix well.
Got it. So lipids are like the rebels of the biomolecule world.
Yeah, they like to do their own thing.
They do their own thing, but what are they made of and what do they actually do in our bodies?
Well, the building blocks of many lipids are fatty acids. They're long chains of carbon and hydrogen with a special group at one end.
Okay.
Now, these fatty acids can be saturated or unsaturated.
Good. I've heard those terms before, but I'm not sure I fully understand the difference.
It all comes down to whether there are double bonds between the carbon atoms in the chain. Saturated fatty acids have no double bonds, so they're straight chains.
Like stiff Lego blocks?
Exactly. Stiff Lego blocks, and unsaturated fatty acids, on the other hand, have at least one double bond, which creates a kink or bend in the chain.
Okay, so like flexible straws.
Yes, flexible straws.
So the saturated fats are all packed in tight, while the unsaturated ones have some kinks and bends from those double bonds.
Exactly, and those structural differences affect their properties in a big way.
Yeah.
Saturated fats like butter are usually solid at room temperature because those straight chains can pack together tightly, but unsaturated fats like olive oil are liquid because those kinks prevent them from packing so closely.
So the shape of the fatty acid chains determines whether a fat is solid or liquid at room temperature. That's fascinating. But why does it matter? Is one type of fat healthier than the other?
That's a great question, and it's a bit more complex than just good fats versus bad fats. We'll dig into that more in a bit.
Okay, so we've got saturated fats, straight, and solid unsaturated fats, kinked and liquid. But what do they actually do in our bodies?
Well, to really understand their roles, we need to talk about two key types: triglycerides and phospholipids.
Ooh, those sound familiar, triglycerides. Those are the fats we store in our bodies, right? And phospholipids, something to do with cell membranes.
You got it. Triglycerides are a main energy storage form. They're made of 3 fatty acids attached to a glycerol molecule. Think of them as little packets of concentrated energy. And phospholipids are the key component of cell membranes. They have 2 fatty acids plus a phosphate group attached to glycerol.
So triglycerides are like our energy reserves and phospholipids are the architects of our cells. That's pretty cool. But wait, there's one more group of lipids we haven't talked about yet. Steroids, what are those all about?
Steroids are fascinating. They have this unique structure with 4 fused carbon rings. Think of hormones like testosterone and estrogen. Those are steroids. And just like other lipids, they're hydrophobic, which means they can easily slip through cell membranes.
Wow, so steroids are like the stealthy ninjas of the lipid world, slipping through membranes with ease.
That's a great analogy.
Yeah.
Now, remember how we talked about phospholipids having a hydrophilic head, which loves water, and hydrophobic tails, which fear water?
Yeah, that was pretty cool. So how does that work in a cell membrane?
Imagine you drop a bunch of phospholipids into water. What do you think happens?
Well, I guess the heads would want to be near the water, and the tails would want to stay as far away from it as possible.
Exactly. They self-assemble into this amazing structure called a phospholipid bilayer.
A phospholipid bilayer.
The hydrophilic heads face outwards toward the watery environments inside and outside the cell, and the hydrophobic tails tuck themselves inwards away from the water, forming a kind of oily core.
So it's like a sandwich with the water-loving bread on the outside and the water-fearing filling on the inside.
Perfect analogy. And this bilayer structure is what makes cell membranes selectively permeable, meaning some things can pass through easily while others can't. It's like a gatekeeper controlling what goes in and out of the cell.
So that's why those sneaky steroids can slip through their hydrophobic, so they can hang out with the fatty acid tails in the middle of the membrane.
Precisely, and that's how many hormones, which are often steroids, can enter cells and deliver their messages.
Wow, so much is going on in those cell membranes. Okay, we've talked about structure, but I'm also curious about energy. Mr. Martin mentioned that lipids are really good at storing energy. Is that true?
Absolutely. In fact, gram for gram lipids store more than twice the energy of carbohydrates. That's why our bodies store excess energy as fat, those triglycerides we talked about.
So if I eat a big plate of pasta, my body might store some of that energy as glycogen. But if I eat a giant slice of cake, it's more likely to end up as fat.
That's a simplified way to think about it. But yes, glycogen is great for short-term energy storage, but when it comes to packing away energy for the long haul, triglycerides are the champions.
Makes sense. And besides storing energy, what other important jobs do lipids have?
Well, as we discussed, they form the basis of cell membranes controlling what enters and exits the cell. They also provide insulation, particularly in animals living in cold environments. Think of blubber keeping whales and seals warm.
Right, like a cozy layer of fat under their skin.
Exactly, and don't forget, those hormones we talked about, many of them are steroids, which are lipids. Hormones regulate everything from growth and development to metabolism and reproduction.
So lipids are involved in energy, cell structure, insulation, and even communication within the body. They're like the multi-talented performers of the biomolecule world.
I like that.
But hold on a second. I remember Mr. Martin mentioning something about saturated and unsaturated fats being different in terms of our health. What's the deal with that?
You're right. It all comes down to the structure of those fatty acid chains. Remember, saturated fatty acids have no double bonds between carbons, so they're straight chains, while unsaturated fatty acids have at least one double bond, creating a kink in the chain.
So saturated fats are like straight, rigid Lego chains, and unsaturated fats are more like bendy straws.
That's a great way to visualize it, and those structural differences affect their properties. Saturated fats tend to be solid at room temperature because those straight chains can pack tightly together, think butter lard or the fat in a juicy steak.
And unsaturated fats are liquid because those kinks prevent them from packing so tightly.
Precisely, think olive oil, avocado oil, or the oils in nuts and seeds.
Okay, that makes sense. But why does it matter for our health? Is one type of fat healthier than the other?
Well, it's not quite that simple. Both saturated and unsaturated fats play roles in our bodies. But in general, diets high in saturated fats have been linked to an increased risk of heart disease.
So maybe go easy on the butter and bacon.
Moderation is key, but remember, unsaturated fats are generally considered healthier, and within unsaturated fats, there are two types, monounsaturated with one double bond and polyunsaturated with multiple double bonds. Both are beneficial, but polyunsaturated fats contain essential fatty acids that our bodies can't produce on their own.
Essential. So we have to get them from our food.
That's right. These essential fatty acids play important roles in brain function. Cell growth and inflammation control omega-3 and omega-6 fatty acids are examples. You'll find them in foods like fatty fish, flax seeds, and walnuts.
So we need a mix of fats. But it's good to emphasize those healthy unsaturated fats, especially those with essential fatty acids.
That's a great takeaway. Now, remember how we said that unsaturated fatty acids have a kink in their chain because of that double bond?
Yeah, the bendy straw effect.
Well, there's actually another layer of complexity there. That double bond can exist in two different configurations, cis and trans.
Do you another cis and trans thing? I thought we were done with those when we talked about glucose isomers.
I know it can be a bit confusing, but these terms have different meanings in the context of fatty acids in a cis configuration. The hydrogen atoms attached to the carbons in the double bond are on the same side of the molecule, which accentuates that bend in the chain. But in a trans configuration, the hydrogens are on opposite sides, making the chain straighter.
So trans fats are kind of like unsaturated fats pretending to be saturated fats.
That's a good way to put it, and unfortunately, trans fats are not our friends. They're primarily created through a process called hydrogenation, which is used to turn liquid oils into solid fats like margarine or shortening.
So those processed foods are sneaking in some unhealthy trans fats.
Exactly. And trans fats have been linked to even greater health risks than saturated fats, including increased LDL cholesterol, the bad kind, and decreased HDL cholesterol, the good kind.
So it's not just about saturated versus unsaturated. It's
Right.
also about avoiding those sneaky trans fats.
Yeah.
Is it getting complex?
It definitely highlights the importance of reading food labels and understanding the different types of fats. Now shifting gears a bit, let's touch on another key difference between carbohydrates and lipids, their role in energy storage.
Okay, we talked about lipids being more efficient at storing energy gram for gram, but why is that?
Well, it has to do with the chemical bonds. Lipids with all those carbon-hydrogen bonds are more reduced than carbohydrates, which means they have more electrons available to transfer during cellular respiration, and that transfer of electrons is what generates energy in the form of ATP.
So more electrons, more energy. It's like
Yeah.
lipids have a higher energy density than carbohydrates.
Precisely, think of it this way. Burning a gram of fat releases more heat than burning a gram of sugar. That's why fats are such a valuable energy source for animals, especially those that need to store large amounts of energy for long periods, like migrating birds or hibernating bears.
Wow, nature is so efficient.
Yeah.
Okay, so we've covered a lot about carbohydrates and lipids, their structures, functions, and even how they impact our health.
And we've only just scratched the surface, but understanding these fundamental concepts is crucial for unraveling the complexities of life.
I completely agree this deep dive has been incredibly helpful. I feel like I have a much better grasp of these essential biomolecules now.
I'm glad to hear that.
It's amazing how everything comes back to like those basic chemical principles. So to recap, we've got carbohydrates from those simple sugars like glucose to the more complex ones like starch and cellulose. They're important for energy structure and even cell signaling.
That's right. And then on the other side, we have lipids, which are hydrophobic and include fats, oils, and even some hormones. They're great at storing energy. They make up those cell membranes and they provide insulation too.
Okay, so we've covered the basics, but let's dive a little deeper into some of the stuff Mr. Martin might ask about on a test.
Okay, sounds good.
For example, he mentioned something about the different levels of protein structure. What's that all about?
Ah, yes, protein structure. It's fascinating. It's like origami, but with molecules.
Origami.
Yeah, imagine you have this long chain of amino acids.
Those are the building blocks of proteins, right?
Exactly, the building blocks. And this chain, it's not just floppy, it folds and twists in these very specific ways to create a 3D structure.
Okay, so it's not just a random tangle of amino acids. There's an actual shape involved.
Precisely. And there are 4 main levels of protein structure.
4?
Yeah. The primary structure is simply the sequence of amino acids in the chain, like the order of beads on a string.
So the primary structure is like a list of ingredients.
Yes, a list of ingredients.
And then what?
Then we have the secondary structure, which involves local folding patterns like twists and turns within the chain.
So that's how you start mixing the ingredients together.
I like that analogy.
Okay.
Now, as the chain continues to fold, we get to the tertiary structure,
Tertiary.
which is the overall 3D shape of a single polypeptide chain. It's like taking that mixed dough and shaping it into a loaf of bread.
Okay, so we've gone from a straight chain to twists and turns to a complex 3D shape. Is that the final level?
There's one more, the quaternary structure.
Quaternary.
This one describes how multiple polypeptide chains or subunits come together to form a larger protein complex. It's like taking multiple loaves of bread and arranging them in a basket.
So it's like building a multi-story Lego structure. You start with individual bricks, then you make walls, and then you combine those walls to create a whole building.
Exactly. And just like with a building, the final shape of a protein determines its function.
Okay, so the sequence of amino acids determines how the protein folds, and that final 3D shape determines what the protein actually does. That makes sense. But what forces are actually holding the protein in that specific shape?
That's a great question. There are several types of bonds and interactions that contribute to protein folding. Well, we have hydrogen bonds, which are weak but numerous. They act like tiny magnets holding different parts of the chain together.
Okay.
Then there are ionic bonds, which form between oppositely charged amino acids. And we can't forget about hydrophobic interactions. Remember how lipids hate water?
Yeah, they're hydrophobic.
Well, some amino acids are hydrophobic too, and they tend to cluster together in the interior of the protein away from that watery environment.
So it's like this delicate dance between different forces all working together to create that perfect 3D shape.
Exactly. And if those forces are disrupted, the protein can lose its shape and stop functioning properly.
That's what happens when you cook an egg, right? The heat disrupts the bonds in the egg white proteins, causing them to unfold and solidify.
Precisely. That's a great example of protein denaturation, where the protein loses its structure and function.
Okay, so protein folding is essential for their function. But Mr. Martin also mentioned something about enzymes being a special type of protein. What makes them so special?
Enzymes are like the catalysts of life. They speed up chemical reactions within cells without being consumed themselves.
So they're like the little helpers that make things happen faster.
Exactly. Every cell relies on enzymes for countless processes, from breaking down food for energy to copying DNA. And the amazing thing is that each enzyme is incredibly specific, designed to catalyze a particular reaction.
So it's like a lock and key. Each enzyme has a specific shape that fits a particular molecule, like a key fitting into a lock.
That's a classic analogy, and it's a good way to understand enzyme specificity. The active site of an enzyme is a region with a unique shape that perfectly complements the shape of the molecule it acts on, called the substrate. When the substrate binds to the active site, it forms an enzyme-substrate complex, and that's where the magic
So
happens.
the enzyme grabs onto the substrate, helps it transform into something else, and then releases it to do its job.
Hmm.
And the enzyme itself is unchanged, ready to do it again.
Exactly. And the rate at which enzymes work can be affected by various factors like temperature and pH. Think about what happens when you have a fever. Your enzymes might not work as efficiently because the temperature is too high.
Wow. So enzymes are like tiny machines, precisely designed to speed up the chemical reactions that make life possible.
Yeah.
It's incredible how much is happening at the molecular level.
It really is. And as you continue to explore the world of biology, you'll see just how crucial these biomolecules are for every aspect of life, from the smallest cell to the largest organism.
This deep dive is already giving me a whole new appreciation for the complexity and beauty of the natural world. It's like peeking behind the curtain and seeing all the amazing machinery at work.
Yeah, it really is mind-blowing to think about all these processes happening inside us all the time. We've covered so much ground today, from those building blocks of carbs and lipids to the complexities of protein structure and enzymes.
Yeah, and we've done it in a way that's hopefully easy to understand, even if English isn't your first language.
So remember, Mr. Martin's class, the key is to break down those complex concepts into smaller, more manageable chunks.
Absolutely. Don't get overwhelmed by all the big words and all those intricate diagrams.
Yeah, focus on the main ideas.
And don't be afraid to ask questions.
Yeah, absolutely. And remember, these aren't just abstract ideas, These things are happening inside you right now.
That's so cool.
Every time you eat a meal, every time you move a muscle, you're witnessing those carbohydrates, lipids, proteins, enzymes at work.
It's like our bodies are these incredible self-assembling machines, constantly building and breaking down molecules to keep us going.
Exactly. And as you continue your studies in biology, you'll discover even more fascinating details about these biomolecules and how they interact to create that complex tapestry of life.
Well, I think we've given Mr. Martin's class a pretty solid foundation for understanding, the key structures, functions, and differences between carbohydrates and lipids.
I hope so. And remember, this knowledge isn't just about passing exams. It's about understanding the building blocks of life itself, and how these molecules interact is going to be so important for understanding those more complex biological processes like metabolism, cell signaling, even disease.
Right, it's like learning the alphabet before you can write a novel.
Exactly.
Knowing these fundamentals opens up a whole new world for further exploration.
So keep asking questions, keep exploring, and keep that scientific curiosity alive.
Great advice, and if you have any questions, don't hesitate to ask Mr. Martin. He's a fantastic teacher and an expert in all things biology.
Absolutely, Mr. Martin. We salute you, and your students keep up the great work.
And to everyone listening, thanks for joining us on this deep dive into the world of carbohydrates and lipids. We'll see you next time for another fascinating exploration of the science that makes life possible.